Welcome back, everyone, to our Explainer series on NCERT Class 11 Biology. I'm absolutely thrilled to be guiding you through Chapter 8, Cell, the Unit of Life. Today, we're focusing all our energy on Part 2, Prokaryotic Cells. If you're studying for your Neet exams, well, grab your notes because you're in exactly the right place. We're going to break down the anatomy of these incredible, primitive cells, and we'll do it visually. So let's jump right in.
Real quick, before we zoom into the microscopic world of bacteria, let's look back at Part 1. We established that the cell is the absolute fundamental unit of life. Basically, anything less than a complete cell structure just cannot survive on its own. We also split life into two big buckets: prokaryotes, the ones without a membrane-bound nucleus, and eukaryotes, the ones that have one. And today, the spotlight is 100% on the prokaryotes. To make this stick in your brain for the exams, we're taking an outside-in journey. Number 1, the bacterial cell envelope. Number 2, the surface appendages for survival. And number 3, the cytoplasm and internal structures. Let's start by getting some perspective on how tiny these guys really are. A typical eukaryotic cell, like the ones making up your body right now, is about 10 to 20 micrometers. Now compare that to a typical bacterium. It's just 1 to 2 micrometers. They are incredibly small, which actually allows them to multiply way faster than our cells do. And check out the PPLO, Pleuro Pneumonia Like Organisms. These are mycoplasmas. They're the tiniest cells out there, clocking in at a crazy 0.1 micrometers. Now, even though prokaryotes have a really similar internal setup, their outer shapes, they are all over the place. There are 4 basic shapes you absolutely need to know. First up, bacillus. That's your rod-like shape. Then there's coccus, which is perfectly spherical. Vibrio looks exactly like a little comma. And spirillum, well, just like it sounds, it's spiral-shaped. Make sure you map those visual shapes directly to those exact biological terms. Section 1. The bacterial cell envelope. Let's look at their outermost armor. Most prokaryotic cells, especially bacterial cells, have a chemically complex cell envelope. Think of it as a tightly bound, 3-layered suit of armor. The absolute outermost layer is called the glycocalyx. Right beneath that is the cell wall, and the innermost layer of this envelope is the plasma membrane. Now, even though each layer does its own specific job, it's super important to remember they all team up to act as one single cohesive protective unit. What's really fascinating is how that very first layer, the glycocalyx, can totally change a bacterium's personality. It varies a lot in thickness and makeup from one bacterium to the next. In some, it's just a loose sheath. We call that a slime layer. But in others, it's incredibly thick and tough. When it's built like that, it's called a capsule. This is a very common place to get tripped up, so definitely remember the difference between the loose slime layer and the tough capsule. Because these envelopes can be so different, scientists had to come up with a way to classify them. Enter gram staining. Bacteria are sorted into 2 massive groups based entirely on how their cell envelopes react to a specific stain developed by a scientist named Gram. Gram-positive bacteria take up the stain. Gram-negative, they do not. Let's talk about why those next 2 layers actually matter. The cell wall, that dictates the shape of the cell and gives it the strong structural support it needs to literally not burst or collapse. Moving inward, we hit the plasma membrane. The selectively permeable layer acts as the bouncer, interacting with the outside world. And here's a fun fact. It's actually structurally very similar to the plasma membrane we see in the much more complex eukaryotes. All right, Section 2, surface appendages for survival. Bacteria can be motile, meaning they move around, or non-motile. If they do move, they have these thin filamentous extensions called flagella popping out of their cell wall. A bacterial flagellum has 3 distinct parts: the filament, the hook, and the basal body. That filament is the longest portion, sticking way out into the environment, basically spinning like a microscopic outboard motor. Okay, listen closely, because this right here is a massive trap for biology exams. Besides flagella, bacteria also have these things called pili and fimbria. Pili are elongated tubular structures made of a special protein, and fimbria are these small bristle-like fibers. But here is the kicker. Neither of them have anything to do with motility. 0. They are strictly for attachment. Fimbria, for example, help the bacteria stick to rocks and streams or hold on to host tissues. So just remember, flagella for movement, pili and fimbria for attachment. Time for Section 3, cytoplasm and internal structures. Let's dive straight inside the cell. Once we break through that plasma membrane, we're in the cytoplasm, a fluid matrix filling up the whole cell. But since we're looking at a prokaryote, the biggest thing you'll notice is what's missing. There's no well-defined nucleus. The genetic material is basically naked. There's no nuclear membrane wrapping it up whatsoever. It's just hanging out right there in the cytoplasm. Now, this brings up a really cool question. If their main DNA is just this naked, tangled loop, what gives some bacteria unique survival traits? Like, say, the ability to resist antibiotics?
The secret is extra chromosomal DNA. See, bacteria have their main genomic DNA, which is usually just a single circular chromosome. But lots of them also have these tiny extracircular pieces of DNA floating around called plasmids. These plasmids are essentially biological cheat codes. They give the bacteria unique phenotypic traits, like that antibiotic resistance we just mentioned. They're actually so handy that scientists even use plasmids in the lab to monitor how bacteria take in foreign DNA. Next up is a highly unique feature of prokaryotes called the mesosome. The mesosome is a specialized structure formed when the plasma membrane simply folds and extends inward, right into the cell cytoplasm. These extensions usually look like vesicles, tubules, or lamella.
Lock this into your memory because these are incredibly high-yield facts. Mesosomes are basically multitasking powerhouses. They help form the cell wall, replicate DNA, and pass that DNA along to daughter cells. They also help out with respiration and secretion, plus they massively increase the surface area of the plasma membrane, packing in more enzymes. Oh, and a quick side note, some prokaryotes, like cyanobacteria, have similar membranous extensions called chromatophores, and those contain pigments.
Now, prokaryotes don't have membrane-bound organelles, but they absolutely do have ribosomes for synthesizing proteins. In bacteria, these are attached to the plasma membrane. They're tiny, just about 15 by 20 nanometers. But the magic number you need to memorize is 70S. The prokaryotic ribosome is 70S. It's made of a larger 50S subunit and a smaller 30S subunit. I know 50 plus 30 is 80, but don't let the math freak you out. In the world of sedimentation coefficients, a 50S and a 30S come together to make a 70S. When a bacterium needs to build proteins efficiently, a whole bunch of these ribosomes will latch onto a single strand of messenger RNA, or mRNA. When they form this chain, it's called a polyribosome, or polysome. All the ribosomes on that polysome work together to rapidly translate the mRNA instructions into proteins. Think of it like a highly efficient microscopic assembly line. Lastly, let's talk about where they store their reserve materials. They stash them right in the cytoplasm as inclusion bodies. And here's the key. They aren't wrapped in any membrane system. They're just free-floating. We're talking phosphate granules, cyanophane granules, and glycogen granules. You'll also see gas vacuoles in blue, green, purple, and green photosynthetic bacteria, which literally act like little balloons to help them float. And there you have it. We've built the cell entirely from the outside in. You now have a rock-solid structural grasp of prokaryotic anatomy, from the glycocalyx all the way down to the inclusion bodies. Now that we understand the organization of prokaryotic cells, in the next part, we will move to the more advanced and highly organized eukaryotic cells.